Personal DNA testing, also called genetic testing, is a type of medical test that identifies change in chromosomes, genes, or proteins. The result of a genetic test can confirm or rule out a suspected genetic condition or help determine a person's chance of developing or passing on a genetic disorder. More than 1,000 genetic tests are currently in use, and more are being developed. Several methods can be used for genetic testing. Molecular genetic tests or gene tests study single genes or short lengths of DNA to identify variations or mutations that lead to a genetic disorder. Chromosomal genetic tests analyze whole chromosomes or long lengths of DNA to see if there are large genetic changes, such as an extra copy of a chromosome, that cause a genetic condition. Cloning DNA. DNA cloning is the starting point for many genetic engineering approaches to biotechnology research. Large amounts of DNA are needed for genetic engineering. Multiple copies of a piece of DNA can be made either by using polymerase chain reaction (PCR) or by cloning DNA.
Biological and medical research and discovery. DNA sequences has become indispensable for basic biological research and in numerous applied fields such as medicinal diagnosis, biotechnology, forensic biology, virology, and biological systematics. Genomics is an interdisciplinary field of biology focusing on the structure, function, evolution, mapping, and editing of genomes. A genome is an organism's complete set of DNA, including all of its genes. Genes may direct the production of proteins with the assistance of enzymes and messenger molecules. In turn, proteins make up body structures such as organisms and tissues, as well as control chemical reactions and carry signals between cells. Also involves the sequencing and analysis of genomes through use of high-throughput DNA sequencing.
Plant biotechnology. Plant biotechnology is a set of techniques used to adapt plants for specific needs or opportunities. Situations that combines multiple needs and opportunities are common. For example, a single crop may be required to provide sustainable food and healthful nutrition, protection of the environment, and opportunities for job and income. Finding or developing sustainable plants is typically a highly complex challenge. Plant biotechnologies that assist in developing new variants and traits include genetic and genomics, marker-assisted selection, and transgenic crops. These biotechnologies allows researchers to detect and map genes, discover their function, select for specific genes in genetic resources, and biotech barn yards, knockout cells, and organ factories. Xenotransplantation is the transferring of an organ from one species to another. Pigs can be engineered to lack certain genes that could cause rejection problems when their organs are transplanted to humans.
Safety issues of biotechnology. There are four main concerns we as a society have when it comes to this ever-advancing field. The first one being harm to the environment. This concern is perhaps the most widely cited by those opposed to GMOs. It is very difficult to predict what will happen in an ecosystem where a new organism has been introduced, whether genetically modified or not. Take weeds for example. If farmers introduce a herbicide-resistant marker into a plant, there is the possibility those traits may be transferred to a weed, making it resistant to herbicides as well. The second one being bioterrorism. Governments are worried terrorists will use biotechnology to create new superbugs, infectious viruses, or toxins for which we have no cures. According to the CDC, bioterrorism happens when viruses, bacteria, or other germs are released intentionally to inflict harm or kill people, plants, or livestock. The agency says the most likely agent to be used in an attack is anthrax, a serious disease caused by a bacteria found naturally in soil. The third issue of biotechnology would be laboratory or production safety.
From haystacks to needles. The polymerase chain reaction, or PCR, is now one of the most commonly used methods for obtaining a particular segment of DNA or RNA. It is fast and extremely sensitive. By amplifying a designated segment of DNA, it provides a means to isolate that particular DNA segment or gene. This method requires knowledge of the nucleotide sequence at the end of the regions that you wish to amplify. Once that this is known, one can make large quantities of that region starting with minuscule amounts of material, such as the DNA within a single human hair. With the ability of almost complete or complete sequences of genomes from many species, the range of genes to which it can be applied is enormous.
Also concerned about technicization, safety in laboratories, even under secure conditions when working with organisms of unknown virulence. Another issue would be ethical issues. Besides the age-old debate over whether cloning genes is sacrilegious, innumerable ethical questions arise over the appropriateness of licensing genetic inventions and other IP issues. Genetically modified humans. Human genetic modification is a direct manipulation of the genome using molecular engineering techniques. Recently developed techniques for modifying genes are often called gene editing. Genetic modification can be applied in two very different ways: somatic genetic modification and germline genetic modification. Somatic genetic modification adds, cuts, or changes the genes in some of the cells of an existing person, typically to alleviate a medical condition. Germline genetic modification would change the genes in eggs, sperms, or early embryos, often referred to as inheritable genetic modification or gene editing for reproduction. These alterations would appear in every cell of the person who developed from that gamete or embryo, and also in all subsequent generations.